Well, I do hope that you can hear me now. Yeah, I just realized that basically there was no sound. I can at least hear myself, and I'm quite sure that you can as well. Wrong microphone setting, but at least I got that sorted. Okay, so I'm gonna start again. Hi, hello everyone. I'm Oliver, and welcome to another Microscopy live stream. Okay, yeah. I'm back again. Last week, I a little bit unexpectedly. Well, actually, had to prepare. I had to prepare a video. I was not live because for private reasons. But today I'm again live here, and one of the things that I would like to show you, I'd like to put this under the stereo microscope. There's been a request a couple of weeks ago that I do a little bit more stereo microscopy again, so why not do that? This one I collected just a few hours ago, but you can see it's a little bit dried up already, and yeah, we're gonna put this under the microscope a little bit here. I found on this plant sample, I found a couple of spiders, 2 spiders. I'm also going to look at this. And then, of course, also a little bit of a recap from last week. I want to, again, put the water sample with the ciliates that I've got over here under the microscope to see how that sample has developed. And we're going to look at a few slides that we prepared 2 weeks ago and to see how they are actually looking, because actually their quality has degraded also a little bit. So for this reason, we're going to, yeah, I'm going to do a collection of a variety of different things. As always, a welcome all around the world. I'm not going to go through the chat, but essentially, yeah, hello everyone. And if you have any comments or questions, please feel free to write them in the chat. I will occasionally have a look at the chat and also try to answer some of the questions. So this is stinging nettle, of course, we all know. Look, there's another plant over here. It's not part of it. It's a different one. Of course, we all know that when we touch it, then actually sometimes it can hurt. So why is that? Well, because it stings, obviously, and there is a substance, an acid in there, where basically, yeah, it starts to hurt a little bit. And I'm just going to show it to you directly. The answer to this mystery, which is not really a mystery, I already put it under the microscope. Here it is, and that's already the answer to the question. This is the reason why the stinging nettle stings, because you've got those little spikes, yeah, especially on the stem. Okay, so let's see if we can go in yet further with yeah. No, I'm going to show you the low magnification first, right? So this is how it looks like. Then you can see that over here on the stem and also here on the side of the leaves. Yeah, sometimes you have to go into focus. You're able to see that essentially, yeah, they're quite spiky here, right? So this is basically the reason, and I want to show you something here. Let's take those big ones over here. What actually happens when you touch them? Unfortunately, I was not able to see it this time. It depends a little bit maybe how fresh the plant is, but I've got a pin here, right? Here's a pin, a regular pin as used in sewing. And look at this without here, just for size comparison, right? Look what happens when you touch it. They're pretty flexible, right? Actually, they look kind of soft. And I've done this already before, and then I was able to actually see that there was a droplet of liquid actually came out. Unfortunately, I'm not able to see that here. Okay. But I would like to try the following. I would actually try to cut this off, and I would like to try to make also a slide and to have a look at this using also my compound microscope. Yeah, so essentially what this means is that if you actually pick those stinging nettles, then you actually don't touch it like this, but actually you have to kind of stroke it a little bit and then touch it, because this will then bend over the spikes so that they cannot puncture your skin. Yeah. So this is yeah. You want to have some. I see that some of you want to have some cross sections of the stem, and you know what I say? Why not? Okay, I have no problems. Let's do some cross sections then of the stem as well. I would like to, however, try to put one of those spikes here a little bit under the microscope as well. Okay, what I'm gonna do is right now is I did prepare a short video as well. Here it is. So that this is basically was in the just in the afternoon today. Yeah, behind yeah, where I live in my behind my apartment, there's this yeah, just as a size comparison, how big they actually are. Right, they're almost my size, and what I, however, did is I tried to pick some smaller ones, some fresh ones here, of course, with a scissor because I did not want to actually touch them myself, right? Yeah, and then the video starts to loop again. So sometimes you can actually also see that there are some holes in the stinging nettle over here. You see, and these tiny holes here are usually because of caterpillars or maybe other insects that have actually eaten some of those stinging nettles. Okay, so that's a little bit the yeah, the background here. So when we what we can do is I have to dig out I have to find my microtome again, and here it is. So let's try to again make then later a little cross section of the stinging nettle, which I don't remember that I have ever done before. So I'm just gonna get everything prepared here. Here it is. Okay, got that stuff here ready. You see, I need this here. Remember, and the microtome. But what I would like to do first is and that's going to be maybe the more difficult part. Is I actually would like to put some of those where's this? Yeah, those things here under the microscope. So what I need to do is I somehow need to be able to cut this off carefully, and that's going to be a slight challenge. So I'm going to go back to the desk view. Yeah. So yeah, eating is also possible. Not only that, some people make actually tea. You can actually make you use fresh ones, you dry them, and then you can make tea. Actually, apparently quite healthy. And this is now going to be the big challenge, and I have no idea how I'm going to do that. How am I supposed to cut this off here? Um, those tiny things here. I have really no idea. So what I'm going to try to do is I'm going to simply try to maybe I'm going to grab it here. Do the easy way first. I'm just going to try to, you don't see it here, just take part of the stem here first. Another question is how am I supposed to just get the, I have really, guys, folks, I have really no idea. Okay, but you see over here the very fine, yeah, spikes. And I'm going to maybe try to cut it longitudinally. Okay, and then hope for the best. Here we go. I was looking for my needle again, and I misplaced the needle. Here it is, over here. Okay. Yeah. So, ooh, it's out of focus. Bad. Okay. Yeah. So, and I don't know if this is actually thin enough. I see I kind of cut the stem not longitudinally, because it was too difficult for me to actually get the and then let's hope for the best that this actually will be sufficiently thin that we're actually able to see, yeah, this. I'm now going to do a very quick check without any mounting medium, without any water. I'm just going to put it directly under the microscope. I need to turn on the lights as well. Let's just do a first quick quality check. And actually, this is it. This is actually... look at this, look at this. So this is actually not so bad. Not so bad. Not so bad. So this is without water, without anything, and it's not flat. Therefore, it's, you see, if I focus all the way through here, yeah, it's, this is a tiny droplet maybe here at the very, yeah. Yeah, there seems to be spines on the spines. I don't know if this is, yeah. So what I'm going to do now is I'm going to, of course, going to try to compress this a little bit. This is going to be a little bit difficult because it's fairly thick as a specimen. But let's give it a try in any case. What I need is of course, a little bit of water. So, and then I'm just going to be pretty cruel and brutal. I'm just going to press down on it and I'm going to compress it and hope that, yeah. This will then make sure that this is going to be sufficiently flat. So cover glass goes on top here. You see, it is fairly thick. So I now need a solid thing here, and I'm going to simply press down on it now. Kind of compressing the specimen and hoping that I'm able to see something here. That is really a question of luck now. Here, let's see if we're able to find it. Compression specimens are nothing uncommon. And we were lucky. We were lucky. Gee, am I relieved? Here it is. You see, there is a little bit of a yeah. Over here. Let's see if there's another one over here. This one is actually look. This one is actually quite nice. It came off here, right? So yeah, and here it is. One of those. Yeah, and here you see actually here on the stem there are many more. Yeah, and now you can actually also see the individual cells. Here's a huge one again, the one from before. Yeah. It's actually quite a good thing here. You can also see the individual cells here. So I'm actually quite happy. So what I've done is this is called a compression specimen, right? I've already made it very thin, but then I simply pressed down on it, put a cover glass on top, and this looks, of course, my favorite tissue, the xylem. Yeah, this is okay. I'll just show you a little bit here. I mean, every time we look at plants, you see this, it's a little bit spiral-shaped structure, right? These are the vessels that actually carry water up a plant. Let me turn on, yeah. It's these are actually spiral-shaped, reinforcements in the cell wall. It's called the xylem, and you can actually see those dark areas. That's air that's in it, right? So I'm quite happy to be able to see that. But let's look over here directly also on the surface here. You can also see those little spikes here. Little, yeah. It's quite nice, huh? So there is a comment here. There seem to be two different sizes. Are the large on the stinging ones or both? Basically, I'd have to guess now, but I know that there are the larger ones on the side, and then there are, however, over here, plenty other ones also around it. And I suppose maybe it is just a question um, um, of where they grow. But you see here, this is no, that's not one. Here it starts. Yeah, this is a huge one over here. I mean, it's so different in size that it must be a different kind. Yeah. And something that I don't know, I'm not able to see that here, but maybe there is you're able to see a little bit of a canal on the inside where the liquid, yeah, is actually passed out. No, can't see that quite well here. Yeah. Let's turn on DIC just for the fun of it, to give it a little more color. Look at this. This is actually quite surprisingly good. Yeah, because the refractive difference in refractive index, it actually looks pretty nice. But let's look at the other ones that are now found on the surface here. Where are they? Here's one. Do we focus? Yeah, I mean, they definitely look like different kinds. Yeah, yeah. Ah, look at this. Also fun. Look, you gotta watch very carefully. These are stomates. Do you see those things here? Okay, my mouse is a little bit acting up. Now it goes again. You see this here, right? There's a little tiny hole here. These are stomates, which are openings, which I kind of find interesting because I'm looking right now at a stem, right? You can find those stomates on the bottom side of a leaf. Yeah, I kind of like this. Yeah. So staining might, yeah, exactly. Look also over here. I mean, a couple of years ago, I did make a video, and I was quite lucky because I actually was able to see how there was a little droplet of liquid coming out of some of those spikes. But the thing that fascinates me is how extremely thin or pointed they are, right? Yeah. Again, over here, the nice cell. And now you see also here the xylem pretty nicely. Yeah, the spiral-shaped reinforcements. Yeah. Yeah, so that's pretty, yeah, I'm pretty happy with the result. Yeah. Here also the cells are visible. Oh, look at this one is a little bit bent. Sometimes I feel we have, I don't know. Today, as I was preparing a little bit, I wasn't preparing a lot, but as I was collecting the plants and everything, and doing a couple of first observations, I was kind of wondering myself, man, we've got a weird hobby, being fascinated. But on the other hand, honestly, I consider nature very beautiful. Yeah, and the microscope as a tool to access a completely different view of the environment and nature, I think that's pretty fun. Yeah. Yeah. And look at the green chloroplasts. Yeah. So generally, yeah. You see that is very briefly a nice easy way. You just take a small sample of the plant, and then you could simply compress it, and you kind of squash it flat. Yeah. So without cutting. And this way, we're also able to see quite a bit here. Yeah. So here the cells quite nicely visible. Yeah. Yeah. So let's move on. There has been the request to make a cross section of the stem. This is going to be a painful experience now for me. Yeah. Look at this. Yeah, you can even see the cell organelles here. Ah, that's another one here. Two of them. Look. Yeah. I'm just reading this. Yeah. The hobby you pass on is awesome. Yes, thank you for that. Thank you as well. To me, having TikTok as a hobby is weirder than our microscopy hobby. Honestly, I've been the whole thing with social media is one hand a blessing and a problem. The blessing, of course, is that it's easy nowadays to make videos and to have a community and to kind of share the hobby of microscopy. Oh, there's a firework going on the outside here. Today is Saturday. Sometimes when I don't know if you're able to hear it. Uh, it's not a thunderstorm, but sometimes people are marrying on a Saturday here, and then they have on Saturday evening they have a firework. So maybe you're able to hear that. Yeah. Yeah. So quite. Yeah. I just said the following. This whole thing with social media on one hand is a curse and a blessing. A blessing, of course, because it's not easily possible to share a hobby like microscopy or whatever. But then on the other hand, I've heard that a lot of hobbies these days start to disappear because people are more and more on social media. Yeah. So yeah. So it's yeah. Marriage, yes. Yeah. Every now and then on a Saturday, they have a very short, some people are organizing a very short firework. And uh, there was actually a thunderstorm just a half an hour ago. I was a little bit worried that we're going to have a power shortage, but outage, but nothing like that happened, luckily. So, yeah. So, you know what? Let's try the painful thing. Let's try to make a cross section, okay? Honestly, I have also not made a cross section yet of a stinging nettle. Sometimes it's also one of the reasons why I like doing this live stream is because yeah, I also get to do things that I normally wouldn't do. So what I'm going to do now is I need a solid surface because the surface that I have here is actually a soft plastic writing surface to kind of protect the table and to give it a nicer background color. Yeah. But this also means that I cannot cut anything on here. So I'm going to do it like this. Okay, here we go. Let's remove this here, and I need to put this now into one of those specimen holders. Yeah, if you've joined me already previously, you know that I have 3D printed several of these specimen holders. The two holes here in the center, for those of you who have not seen this yet, the two holes in the center are simply for taking it out of the microtome. You're just going to see in a second how this works here. And I've got 1, 2, 3, 4 different diameters for different stem sizes. And I'll be using, I don't know, maybe this one over here. Yeah, this one here on the side. And I think this one actually might go in here. Yeah, see, it's able to fit without problem. And I'm going to take over here a pin. I'm going to push it all the way through to hold it in place. And I hope that I'm able to do that. Because so here we go. Oh, this is here we go. Yes. All the way in, all the way in, all the way in. I don't know. This is a problem I just realized because this pin head is still sticking out. This is bad. So I won't be able to get it. So I won't be able to fit it into the microtome. That's a bad thing here. So what I'm gonna do, maybe I'm going to try to switch this over to the other one. It's a little bit larger. So let's try this here. Let's put this in here, push this all the way in. Yeah, it's not the pin is now not long enough. Okay, so this is a little bit of an issue. Not to worry. I'm just going to put it back into this one over here. No, which one was it? This one over here, maybe. No, let's do this one. It's a little bit, yeah, it's a little bit tighter. And I'll put it now into the microtome. It fell out. I'm going to turn it. I'm not going to clamp it to my table. Okay, I have to turn this, I have to lower the piston here. By turning it, I'm lowering this piston. Okay. So, let's make it a little smaller. Brighter. So, this one goes in here. So, here we go. Going to put it carefully in here. Now, it is definitely too deep inside. Yeah. So, let's, I'm turning it now. And it's slowly being pushed out. So maybe I can keep it like this here. I need my sharp knife, and let's see if the first cut is always a problem. I see it's a tiny amount. And I need also a little bit of water because those cross sections, they dry out very quickly. So the water is up here. And then, so let's see if I'm able to get this here. This was too thin. Yeah, also too thin. Okay, maybe this actually worked here. Quickly into the water. You know what? No, I'm not going to put it into the water down here. I'm going to actually put it directly on a microscope slide. I think we're just going to save the little intermediate step here. So I would say let's do it just like last week. I'm just going to save this intermediate step and let's add the water directly. And let's put this in here. Okay, maybe this works better. And yes, I already mentioned before that the success of this is a question of trial and error. It's too thin. And I will also tell you something else that I've been thinking about. Not had the time yet to try that, but I'm just throwing it out to you. I've been thinking about actually maybe it is somehow possible. I know there are these knives, the ultrasound knives. Basically, they have some kind of a piezoelectric thing so that the knife is vibrating. Basically kitchen knives. They're pretty expensive. And I was kind of wondering if it's also not possible to add some kind of a vibrating mechanism like an ultrasound mechanism to one of these things. You're like some kind of a piezo quartz or something like that, which causes it to vibrate to cut it more easily. Just an idea. Yeah. Yeah. So here, that's another one. Here we go. I'm just gonna make another cut. Yeah, this one was not so nice. And I think I'm just going to, yes, leave it then as it is. Again, in the context of a live stream like this, I cannot take the same amount of time that I normally usually would take. It's a little bit more like a quick demonstration of what's possible. And yeah, sometimes the quality is not the best. Yeah, yeah. Look at this here. Do you actually notice how dirty this one is? There's a reason to that, because I actually washed those cover glasses, but there is some deposit of calcium carbonate. Some water stains are still on here, so I'm going to quickly also wipe it. Yeah. Simply to make it a little bit cleaner. I generally try to reuse as many of the cover glasses and slides as possible. So let's drop it on here. Yeah, still very thick, right? So I do need more water. Let capillary action do the rest. Okay, let's give it a short try. So the specimen is fairly thick, unfortunately. Okay, so let's start off with a low magnification, and it's 10 times. I need to make it a little bit. Here we go. Yeah, of course, as always, there are some air bubbles. Ah, look at this. Are we not lucky? Look, yeah. Look, now you can also see those spikes here on the side. That's actually quite nice. Yeah. This one here is a fairly good cross section. Look. And here on the side, we're able to see those spikes again. Yeah, of course, making now a permanent slide of this would be quite nice, huh? Yeah, the quartz crystal, it's on the microtome will vibrate on a micro and cut better. That's actually, this was kind of my idea, some kind of a quartz or piezo quartz or something like this. Yeah. So this is a little bit something that is worth trying. Yep. Here we go. But maybe look, do you actually see this here? There is indeed this might actually show that it's a hollow. Let me turn on the arrow again. Usually that's the reason why air bubbles sometimes are kind of useful, because this air bubble over here means that probably this spine here this spike is a little bit hollow, right? Also over here is an air bubble. Yeah, it's this irritant. I think if I remember correctly, isn't it formic acid? Look here, also very nice. I love it. I love it. I love it. I love it. What do I see here? What is this here? Let's open it a little bit. Of course. Ah, monocot or dicot. Yes, a dicot. I will explain why. Look at this. This here. That's of course xylem, right? Yeah. Yeah, that's of course xylem. Yeah. So, and let's talk a little bit about this monocot dicot thingy. You see the two types of flowering plants are either the monocotyledons or the dicotyledous plants. The cotyledons are the first leaves that are formed. And this is clearly a dicot plant. Why? Because the vascular bundles, basically the xylem and the phloem, are arranged in a nice circular pattern. So yeah, so you see those vascular bundles here? Again, my mouse is acting up a little bit. Maybe the battery, I've got a wireless mouse. These are the vascular bundles. You see they're kind of arranged in a ring shaped here. Yeah, that's very typical of a dicot plant. While in a monocot, for example, onions and lilies and so on, they have those vascular bundles distributed right around throughout the stem. And they're not arranged in this nice pattern. Yeah. So that's actually one reason why it's a dicot. And yet another one, obviously, is over here. We just look at the vascular. You see, it's branched, right? That's also very typical of a dicot. While, for example, corn, yeah, and grasses and wheat and the flowers, lilies, they have all of those veins that are basically in parallel. Right, so that's why I have these long, elongated leaves. Yeah, these are all monocots. Right, and if you've got those branched venations, basically for transporting the water, and it's also dicot. Yeah, yeah, histamine. Yeah, formic acid. Yes, yes. Yeah, yeah, histamine that causes this immune reaction. Yeah. Yeah, I was just reading a little bit here. Yeah. Ah, okay. I sent you some image of my upgrade through email. Thank you very much. Did not check yet. Yeah, yeah. Kieselsäure. Ah, okay. Salicylic acid. Uh-huh. Yeah, maybe it is maybe it's indeed a combination of several things. Could be. So yeah. So this is again yeah, a very nice yeah, more or less. Yeah. Yeah. I think in any case more or less instructive. And again, we're able to see, and this is the thing that kind of makes me happy, that the cross sections that you're able to make with this microtome indeed are thin enough that you are actually able to see individual cells in a way that they don't overlap. I just show this to you again. If I focus through here, you see it's blurry, out of focus, then it goes in focus, but it's basically still the same cell walls. Yeah. So this means that yeah, it's probably around one, maximum maybe two cell layers thick. So I'm quite happy that the cross section is fairly yeah, not only consistent but also fairly thin. Let me turn let's move the arrow away here again a little bit so that it doesn't disturb too much. Bubble wrap, yes. Yeah. Again, look here again. The xylem, it kind of shows how much stronger it is because it's almost like a wire, right? So yeah, yeah. So what time is it? Okay, half an hour now. I'm going to slowly maybe move ahead a little bit. I do want to show you just for the fun of it. Yeah, I'm just going to show you what else I found now under the stereo microscope. Let me turn on the stereo microscope. Basically, together with let me always start with a low magnification. Together with the plant I found, where is it? I found this here, and of course I had to immediately have a look at it. There are two spiders in here. This one is actually not looking at me. Yeah, they were running around just a minute ago. Yeah, with its basically. Yeah, that's what you. Yeah, so that's the smaller one. Actually, I was hoping to see the eyes of it because, of course, spiders have lens eyes, and somewhere over here is the other one. Yeah, I've got a lid on top now so that they can't run away. Yeah. Maybe I'm just also going to. Here, this one is now running. No, it's crawling up all the time. No, this one here is now over here. Let's see if it starts to. Yeah, so this is also, yeah, something that I like to do every now and then. Sometimes I have occasional discoveries like those spiders or sometimes also insects and so on. And then I like to simply put them under the microscope and observe them a little bit. but somehow instinctively they, here we all see the eyes. Up, and it fell off. Yeah, the sides of the petri dish are quite slippery. To a certain extent, I almost not dare to say it. To a certain extent, they do look kind of cute. Um, I don't like spiders particularly, obviously, but when you actually magnify, when you put spiders under the stereo microscope, they look less scary than some of the insects. Some of the insects with their, I don't know, mandibles and big antennae and so on look actually more like aliens. Spiders actually, and scary. Spiders actually look less scary when you magnify them. I think. No, yeah, yeah. The thing is the following. When we're doing some okay, spiders are not insects, but you get the idea. When we do some insect observations with the students in school, when we catch insects and also put them under the microscope, um, yeah, of course, students at the beginning, they're of course disgusted and yeah, but at the same time, there is this huge fascination, still of the complexity and the beauty, yeah, of those animals, yeah, because if you just zoom in, it's yeah, incredibly complex, yeah, and that's also one of the things that fascinates me, yeah. Peacock spiders, yeah. So yeah, it's quite interesting. And you know what I'm going to try to do in any case? I'm going to remove the lid now, and this should give us a slightly better image quality. Of course, they might escape, but it's okay. Yeah. So, here it is. Yeah, it's trying to escape. No, it didn't. Let's go down with the magnification again. And the two spiders seem to be ignoring each other. I've heard that sometimes if two spiders encounter each other, they might actually not like that. They might not like each other. Let's look for the other one. Yeah, it's over there. Yeah, you get the idea. Okay. So what I'll be doing, it's difficult to find out. What I'll be doing is, of course, I'll be trying to, I'll be releasing them again, obviously. Yeah, but it was kind of a bycatcher with the plant. Yeah. Yeah. So that's every now and then when they start resting. This guy is a little bit. Yeah. Okay. I think enough. Yeah. It's moving objects are always a little bit difficult to observe, obviously. So let's put a lid again on top, and here we are again. Okay. So what I want to do now is maybe look at a few slides from last week. Okay. No, not yet. I'll have a quick look of what some of the comments are. Okay, the brand of my stereo microscope. Okay, I'll tell you the brand. It's a Euromex, and the model is Nexius Zoom. However, it's like this that many stereo microscopes are actually quite similar to each other. Yeah. How long can you keep a semi-permanent slide of onion plasmolysis? Semi-permanent. Honestly, if you want to keep slides for a longer time, you have to make sure that they're free of water. And as soon as water is a little bit present, like semi-permanent, there is always the danger that it's going to start decomposing. That's the main problem. As soon as the moisture is present, then this can actually mean maybe fungi will start to grow. It might start decomposing. So yeah, so difficult to say. It depends a little bit on the storage conditions, but it's limited in time. Yeah. Yeah, so I will quickly have a look. There are not so many questions for me. Yeah. How long would it take? Can you cut them with can you clean them? Uh, I'm, I don't quite know what. Can you clean them with isopropanol as well? Oh, you mean that the slides that I've been cleaning? Okay, or the cover glasses. I think this refers a little bit because I've been cleaning. Generally, it's like this that if your question was about the cover glasses, if you've got those water stains on some of those, like this one, these are some pretty bad ones, right? Then this is actually calcium carbonate. And then you have to use acid, like for example, vinegar or something like that. But isopropanol is, or any other alcohol, is only going to be useful if you've got fats and oils. But generally, there are not so many unless you've got really fatty fingers or something like that, or greasy fingers. It's not going to, isopropanol is probably not going to remove water stains very well. Yeah, it's actually more suitable if you use, let's say, vinegar or other weak acids. Yeah. Um, there are, yeah. Do you run the cover glasses also in the dishwasher? The short answer is no, because they're going to be flying around. However, what I have done is the following, and I'm also doing that with the other slides. I don't put them directly into the dishwasher, but I put the slides into a plastic box with hot water and then some dishwashing powder. So while I don't use the dishwasher itself, I will use dishwashing powder, and I found this to be very effective. Okay, and then of course I'm rinsing each slide individually. Yeah, and it could maybe be that I also did not rinse those properly. Maybe there's even some dishwashing powder left on here. Not a good idea. It's bad for the microorganisms. Okay, so yeah. Yeah, exactly. Lime skill, it's called. This, yeah, calcium carbonate. So what I'm going to do now is the following. I'm going to give you a quick run through through a couple of other slides, the ones that we made before, at least some of them. Some of them are kind of 2 weeks old or so, and I've made them just to show them here. And I'm not going to spend a lot of time on them. It's just a very quick check. So I've made some cross sections of Elodia, and I've mounted them in glycerin gelatin. And indeed, the specimen degraded. I'm just going to show that to you. And then I think 2 or 3 weeks ago, what I've done is I've tried different mounting media as well. Over here on algae. So this one is there's a glue that's Elmer's PVA glue, and that's also glycerin gelatin. So I tried different mounting media as well. And we're going to have a quick look here, because specifically disappointing is the leaf of Elodia, which was actually quite nice and green and crisp, but kind of degraded now a little bit. I just want to show this to you. So it kind of shows that not all permanent slides are made for eternity, unfortunately. So there's lots of dirt here. Yeah. So, and the thing that you might notice is that essentially, let's remove the DIC, it significantly lost in color. Yeah, I mean, it's still a little bit green, but by far not as green as it used to be. And it almost looks a little bit like the contents of the cells has shrunk a little bit. So it does not look quite as fresh anymore and as crisp. Yeah. So this basically, yes, I labeled the slides. So this kind of shows that, yeah, even though it's only about 2 weeks old, it already started to, yeah, to lose a little bit of quality. Yeah. So if you remember that the chloroplasts were really nicely visible, and now they're kind of very deformed, barely visible. You know, so that's one thing. Over here, what is this here? The cross sections of the water plant, the stem cross section. Let's find those. You always start with a low magnification, maybe. Here we go. What's this? The stem cross section? Yeah, let's try to find another one over here. This one over here. And maybe you notice something else. What else do you notice? Okay, a lot of bubbles, obviously. But there is something else that you might notice, and that is the color. You actually notice now that it looks kind of reddish, purplish, pinkish actually. Yeah, and the reason why that is because the glycerin gelatin had a stain added. And the stain now had enough time to actually stain the plant. So because this glycerin gelatin I've actually bought to mount pollen grains, to also stain the pollen, and now you can see that it actually also stained, yeah, the cross section here. as well. So that's why it kind of turned a little bit reddish in color. Yeah. And look at all of those bubbles that started to appear now. Yeah. So that basically means that there is indeed a somewhat of a change in specimen stability. And here now, this is basically, these were now the algae, which I think these were the algae which I mounted in different types of glue, water-based glue. And let's see here again, I have to always adjust this. Yeah, it's kind of very dirty. Here we go. Yeah. So we have to go up with, let's try to find a place where the density is a little bit higher. Here it is. So let's go up with the magnification again. This, by the way, this is again bright field, so no optical staining here. Let's go up yet further. Those string-like structures, many of them cyanobacteria. But if you actually remember a little bit of how it looked like 2 weeks ago with the fresh sample, yeah, no comparison. The fresh sample is so much nicer and better than this one. So this basically means that even here we have a little bit of a degrading of of specimen quality. Let's try the other glue over here. Okay. Yeah, here this one seems to be a little bit, of course, depends also on the specimen concentration. Yeah. But it's also a water-based glue. It's Elmer's PVA glue. And let's now try if I'm able to find it. Now this one here is again glycerin gelatin. Yeah. And you can see that the contrast, yeah, is also a little bit different here. Yeah. So you see that a lot of it, again, is experimentation. But generally, I would recommend, I mean, opinions vary. I would probably recommend that making permanently mounted slides of algae and water organisms. Of course, please try it. But yeah, it's very static. There is no movement. And actually, the thing that makes it so much more interesting is actually the movement that's going on. Yeah, and of course, we don't see that. Yeah, Stenodesmus exactly. Let's zoom in. Stenodesmus is still recognizable. So basically, but again, much nicer in a fresh sample. And this one over here, these are basically, yeah, a mini colony, and there are those 4 extensions here. Yeah, very often they appear in colonies of 4 cells. Yeah. But the many other cells here in the background, they start to kind of lose their color. They start to bleach a little bit. Nothing unusual because, of course, pigments very often start to bleach. Yeah. I think it was a couple of weeks ago when I showed you some slides that were over 30 years old. They were almost completely free of color. Yeah. Okay, just yeah, a little bit of a, how do you say, yeah, evaluation on how the slide quality actually is. And what I'm going to do now, of course, as well, is I'd like to have now also a little bit of a check on how the enrichment culture is doing. Okay, because again, remember, I think 2 or 3 weeks ago, I added some wheat grains into this water sample, and it's starting to look very disgusting. This is actually a good sign. Yeah. So for those of you who were not there when I've done that, if you what I have done is I've taken, these are wheat grains, right? Cereals, in other words, and I have taken 1 or 2 of them and I crushed them between 2 spoons and I simply dropped it into a water sample. And what will happen then is that ciliates and flagellates and paramecia and hopefully rotifers, they will start to grow here. It looks very cloudy. This basically means it's there are probably also plenty of bacteria present. And for this reason, I'm going to now take a small sample and I would like to check now how this actually yeah, looks like under the microscope. Because every yeah, there's a progression of organisms, and every week it might look a little bit different. So what I'm going to do now, because for somewhat a little bit deeper down, actually I can try to use this here as well. So, and then of course I want to be a little bit careful because there are of course bacteria and all this stuff, on the tip. I'm going to actually go directly into here. I didn't catch anything. I don't know if I actually caught a lot. No. I think the problem is that the tip is a little bit too small in diameter. I mean, there will be already plenty of stuff in here, but I was actually hoping maybe. What I'm going to do now is the following. I'm going to take a slightly larger pipette, and I'm going to take a slightly larger sample. This is pretty disgusting, and that's nice. Ooh, it is too much. Way too much. Ah. Won't come off. Here, now it's off. I have a problem. The problem is that it's way too much. So I'm going to now do not good. I need to use a fairly large cover glass. That's not the problem, but it's still too much liquid in here. Where are my large cover glasses? Here they are. And I need to remove quite a bit of the liquid, otherwise. And if you remember also when I started to make the sample, yeah, there were very few there was very few green stuff in there. So also the algae and whatever not started to grow here. And I'm now going to remove quite a bit of this liquid again. I'm going to put it back into the jar because otherwise it's going to be way too flooded. So let's see how this works. Yeah, we have to throw away the tip. And let's give it a try now. Yup, I think this should be okay. There is no liquid spilling out. And yeah, let's see what we're able to find here. Okay, this is now at 4 times with a 4 times objective. Yeah, there's some movement going on here. Let's go up with the magnification here. Yeah, so the sample is quite healthy and alive. Again, a little bit my disappointment is I was hoping to find more ciliates, like or paramecia specifically, which are quite large. But over here we've got other specimens. Lots of rotifers, really. Wow, lots of rotifers. Yeah, so it really depends on what organisms are present in the first place, and then you're simply enriching those. So with a different water sample, you might have gotten different ones. Actually, this is quite cute. Lots of rotifers here. Yeah. whole colony or family of them. Look at this. Yeah, so the specimen, yeah, the microscope slide is also sufficiently thin so that they're all pretty much in focus. Let's remove the arrow again, it disturbs me. Let's add a little bit of color. Let's see if this actually makes a difference. No, not so much. Yeah. But you actually now see how green the algae and so on, how green they are, yeah, and how much color they have lost, yeah, in the permanent slide. What are those circular shaped rotifers? I'd have to check my identification book. Yeah, what they could be. Those lines that you see in the background, those fine lines are most likely cyanobacteria, obviously. And here they are all cramped together, having a more or less happy life, not knowing that they're now being watched by, I don't know, how many people? A lot of people. They have other things to worry about. Yeah, again, the algae over here. So let's have a look again at a lower magnification a little bit and what we're able, if they're able, you have to turn it down, this whole thing here. So that's why I always recommend, if you have problems finding interesting things, take some solid material. Very often, living things are actually next to some debris and solid material. Oliver, I'm just reading the comments here. Oliver, when making permanent slides of rotifers, it's difficult that when they feel the slide is drying out, they enter to a phase that they change their shapes into balls and it's kind of unrecognizable. Yeah. Yeah, so this is generally a problem, and I also uploaded very recently, a couple of days ago, another video where I tried to make a permanently mounted slide of a sample like this, and you have to dry it, and then of course the organisms, they lose their shape. Yeah. Yeah. Off topic, don't know if you saw it. I recently did a 70+ hour time lapse of the purple amoebas. Highly recommend you to check it out when you have some free time. I suppose it's on your YouTube channel. Then I'll check it out. Okay, thank you for the tip. Yeah. So, what else is there? Yes. Could this be here? I know. I wonder. Look, let's zoom in here. Let's zoom in here. This is a circle in the center. So right in the center. I have a suspicion, and my suspicion might be actually correct, that we're looking at an amoeba here. Yep. Yeah. So, and 60 times. Yes. It's actually quite nice. Yeah. So this seems to be an amoeba. You can actually see what it has eaten. Yeah. Yeah, but yeah. So it should be actually able to change its shape. Yeah. Um, yeah. So yeah, you see it's kind of those amoeba are actually quite nice to make time-lapse videos of because then, and when you speed up the movement, then actually you can actually see it much better how it starts to crawl along. But that is of course quite nicely visible. Yeah. Yeah, it's yes, exactly. It seems to have eaten quite something quite big. Yeah. So usually what they do is they take on the color of the food that they eat. So if you see that there's something green inside the amoeba, it's most likely because they've eaten some green algae. Yeah. Yeah. So this is usually amoeba are more difficult to find sometimes because they're easier to overlook because they don't move quite as quickly. Yeah. So let's go down again with the yeah. Yeah, let's what else are we able to find here? Of course, rotifers. Yeah. I mean, again, what I recommend is you just gotta try. Every time when I've looked at water samples and if I made an enrichment culture, I found different organisms. It really depends what's present in the first place. So this here is also quite nice and interesting to see. Gosh, the dark, it's too dark, it's too dark. Okay. So this is actually also something that you see quite frequently here. Again, where is the arrow? This thing here, again, of course, an amoeba, right? Not moving very quickly, but this one is quite interesting. Those tiny little things here, these are bacteria, of course. And this one here is green. So that's an algae and producing oxygen. And very often you see that you will then have bacteria gathering around algae because they need oxygen. Something you can see that quite often. And forgot the name of the scientist, but this was actually a a very remarkable experiment that what has been done back in the day. They've used different colors of light to shine it on those algae. And depending on the color of light that was used, of course, they produced different amounts of oxygen. And they were able to kind of measure the amount of oxygen produced by counting the bacteria or ciliates or whatever which gathered around them. Look, here's a rotifer. Yeah. So kind of the number of organisms gathering around it is kind of like a measure on how much oxygen it produces. Yeah. So let me see. I'm checking some of this. So let me see. I don't have any channel I sent to all of it. Okay, thank you. You sent it. I have to check my email. Okay. I have to check my email then. The 60 times was such an improvement to observing. Thanks for the idea. So it's 20 times. Yeah, this is refers to the following. Many microscopes, when you buy them, they come with 100 times oil immersion objective. And I find it personally not to be, I mean, I'm using oil immersion, obviously, but I don't find it quite as useful as, for example, at 20 times or 60 times. Because the increase in magnification from 60 to 100 is not that big, but you have to have oil and it's messy and so on. Yeah. Yes, this is actually also quite nice, indeed, the size comparison. Yeah. What we have over here is, I mean, cell sizes have a quite a range difference, but you see over here an amoeba, it's a eukaryote with a nucleus, right? The bacteria here, prokaryotes, yeah. The algae over here, also eukaryotes, and maybe if you just see the size difference of the cells, and this kind of makes it much more and the rotifer, which is made of, I don't know, around 1,000 cells, right? So the idea is a little bit the following, that scientists have calculated you've got more bacteria growing on your body and in your body than you have body cells. It's kind of difficult to imagine where are all those bacteria. Well, that's only possible because the bacteria are so much smaller. Yeah. So yeah, those bacteria are simply so approximately 1/1000th of the volume of a eukaryotic cell. Yeah. This one over here seems to be a ciliate, because you can see the tiny little hair, the cilia on the around it. Occasionally, if you're lucky, you also see flagellates. This here, this looks like a starch grain from the. This is a starch grain from the cereal, from the wheat. And of course, again here, see, synedesmas or related, and bacteria swarming around it. Yeah. Also quite so here is, oh, look at this here as well. They sometimes you see this happening after some time when the oxygen kind of level kind of drops. Yeah. A few dozen grams, yes. You can do it the following, a rough estimate. You divide your body mass by 1,000. Okay. That's a very rough estimate because yeah, if you assume that there are now probably as many bacteria as you have bacterial cells as you have body cells, and if you estimate that one bacterial cell has 1/1000th the volume of a body cell, because the diameter is approximately 10 times as small, length times width times height, 10 times 10 times 10 makes a 1/1000th. So then basically, if you take your body mass and divide it by 1,000, then yeah, basically you've got the approximate number of grams of bacteria. And if you feel that this is, if you wonder where do all of the bacteria live, well, then I will tell you most of them in your intestine. Yeah, so the intestine is actually quite full of bacteria. Here again, also very nice. Look, this is actually pretty instructive as well. Yeah. Let me remove this here again. Okay. So let's go down with the magnification. Maybe we're able to find something else that's interesting. Look, this one over here. Yeah. What else is there? Yes. Oh, yes. This is also quite common. Very common as well. Let's be a little bit patient now. Do you actually see that there is over here almost like a, a road of bacteria? And what will normally happen is that they will start to move into one direction together over the course of a couple of minutes. The reason is they will move towards the edge of the cover glass where there is more oxygen present. So they're kind of traveling yeah, together, yeah, towards the side. And we can, I don't know, I'll put right now. It's right in the middle of the yeah, of the microscope of the image. Yeah, I printed some of your magazines. Yes, okay. They follow a gradient. Yes, they follow an oxygen gradient. You have to be a little bit patient here. Yeah, I need to talk a little bit about my website, which was and the forum, which was kind of frustrating the last couple of weeks again. It was maybe some of you have already realized again it was not accessible. Yeah, there were some technical issues which I got sorted out. Yeah, so just in case you're wondering, and what is this bacterial line? I don't know. Came up with the term bacterial front, like a weather's cold hot front. Yeah, so it's some kind of a yeah. It would be kind of interesting to actually come up with a term here. Again, this depends. You're going to be patient to be able to actually see them move, and I've made already some time lapse videos of this as well. Yeah, and you see how the all of those other ciliates and rotifers and so on kind of diving in there and kind of feeding. It's a feeding frenzy. Yeah, yeah. Here we go. Here. So the front goes on, continues over here. Oh, this is quite a bit going on here. But at the same time, I do have to tell you, I mean, while it's, yeah, I mean, the bacterial concentration is pretty high. Yeah, I mean, I mean, those bacteria are from the water, right? So it would be something different than if you were to grow bacteria that you've isolated from a human body or from animals, right? But still, you don't know which bacteria they are, right? So you gotta be still stick to basic hygienics and yeah. What I'll be doing, I'll be rinsing everything properly later on, but the bacterial concentration is fairly high, right? So just be aware of that. Yeah. Ah, yeah. Some people started to order microscopes. Yeah. You got admission for biotechnology. Well, I congratulate. Yeah. A little bit about my personal background, a little bit. Oh, I love this. Look at this. Many of you, of course, know that I've been out for many years. I've been teaching biology in high school. Sometimes a little bit dry, I have to tell you, because the students have to know the facts and the theory. And but originally, I've got a yeah, I've got a training as a microbiologist, and the thing that I found a little bit interesting at that time when I studied microbiology is that it was essentially we did not use microscopes as often as I was hoping to use them because microbiology was actually yeah, mostly a lot of chemistry indeed. So the fascination with living things. Yeah, I had to basically maintain that a little bit by becoming active myself and buying myself a microscope and doing a little bit of hands-on, yeah, activities here. Yeah, um, yeah, yeah, biology book. Yeah, what's the topic that you have to do? You've got tomorrow. You've got a test. So what's the chapter? Yeah. Yeah, but again, I think what's something that's really important is um, something that I think is really important when doing science and so on. Science can become very, how shall I say, dry very quickly, and sometimes, especially if you're doing laboratory work with biology, it's very easy that sometimes you lose a little bit connection to hands-on biology. Yeah. I encourage you to not forget about that aspect as well. Yeah. Um, you can't center the condenser. May I say the following? With a Swift 380T, it is possible to center the condenser. However, if it's a little bit off center, but it's not something that you can do dynamically. There are 3 screws that you can loosen at the condenser, and then you can move it around a little bit. So the holes are a little bit larger so that you can actually adjust the center, and then you can tighten it again. So in that sense, you can center it, but there is no possibility to dynamically adjust the center of the condenser. So if it's a little bit off center, you can actually, at least the model that I have of the 380T, you can actually loosen the screws and make small adjustments. It takes some patience, however. Yeah. Why don't you? This is an interesting question here. Why don't you teach biology on YouTube, like concepts and all? Honestly, I've been thinking about that. And honestly, I've actually made several years ago for my students, I've actually made YouTube videos, which I occasionally show to students. However, teaching biology on YouTube as videos can be a pretty dry thing. So it's mostly theory. Basically, I'll be explaining concepts, making drawings. Students have to take notes. So it's not really like so much fun at looking at things, but essentially I have to explain the structure of a cell membrane, right? And then I have to make drawings. So it's not as quite as hands-on. I've been thinking about that for sure, because there are lots of biology students out there who would probably need that. But it's probably not, how do you say, designed for the masses, right? It's biology theory. Yeah. Could you briefly recap who's eating who in such a sample? Well, I can. Very briefly. Let's go down with the magnification to get a better overview here. Yeah. So what we're able to see is the following. Let's explain this. Not only who is eating who, but what's actually going on here. So what do you have over here? This here is an air bubble because the slide is slowly drying up. And this basically means that those rotifers, which are actively moving and which need a lot of oxygen, they will, of course, like to be next to the air, right? Because the oxygen diffuses into the water, and then they have oxygen here. Over here, what you see over here is you see those bacteria, the bacterial front, as we call it, and it also moves towards the oxygen, right? However, those bacteria are actually being eaten by the rotifers as well. So what we have is we have actually a small food chain. Those little white dots that you see here, these might be some of the starch grains from the wheat that I added as a food. The bacteria eat the starch, and then the ciliates and the rotifers will eat the bacteria. And those rotifers here are not single-celled. They're actually microanimals. They're real animals. So they are multicellular animals. Yeah. Actually one of the smallest animals that there are. And they are also known to be so-called eutelic. And eutelic means that the number of cells that they have is fixed. For example, humans are not eutelic. Different human beings have different number of cells. If you're larger, you have more cells. If you're smaller, you have fewer cells, obviously, right? But with those rotifers and other microanimals, also, for example, tardigrades, they're eutelic, and this basically means that it's possible, and scientists have done that, to trace every individual cell back to the first original cell. Yeah, and they're all female. That's also correct. And this caused a big headache to some scientists. How can they survive if they are all female? What about the genetic variability, the genetic diversity? Why are they able to survive? Because you need actually, normally, in the quotation marks, you need a fertilization to happen to increase genetic variability, diversity. So they're kind of wondering, scientists were wondering, how is it possible if they're genetically all the same? Because they are born by so-called parthenogenesis, which means that they lay eggs, but they're not fertilized. It's a form of asexual reproduction. So if they're genetically the same, how are they able to survive if the environment changes, right? a big mystery because actually they found a solution. It's kind of obvious. What they do is they do horizontal gene transfer. This means is that they have a method somehow to exchange DNA directly with each other to increase the genetics variability. Yeah, yeah. Um, yeah, I get it. I get it. Yeah, is recentering. I get it. Is not practical. Yeah. However, the good news is you don't lose a huge amount of image quality if it's not in the center. Yeah, yeah. I feel kind of sad killing rotifers when trying to make slides, and I know what you're talking about, and I get it. However, yeah, those animals, as far as we know, they don't have a consciousness because they don't have a fully developed brain. Yeah, yeah. The dark field effect. I get it. Yeah, yeah, yeah. So look, this is a real feeding frenzy going on here, or oxygen. Yeah, yeah. So it kind of means that sometimes it's not necessary to slow them down because there's so many of them actually together that it's possible to directly observe them. Yeah. So let's look. I'm gonna put the DIC away. Uh, no, that's no, that's wrong. This one here. That's not bright field. Yeah, fascinating. I mean, I don't know. I've been doing this for so many years. I'm still always fascinated by this. Yeah, yeah, yeah. Exactly. Yeah, I'm reading again. Yes, that's another thing here. I use Google Photos. Generally, that's something I recommend in any case that you can significantly improve, and maybe that's the biggest improvement is you just do a contrast improvement of whatever, yeah, micro photo micrograph you've got. If you do a contrast improvement, then subjectively the image will also appear to me more crisp and sharp. Yeah. What time is it? Yeah, it's 1 minute and 1 hour and 17. Yeah, I think I'm just going to slowly call it a day again, right? Um, yeah, and I have actually read on Reddit today a new slide preparation technique that I would like to experiment with. And that is how it's possible. And somebody posted actually a nice image on Reddit in the microscopy subreddit. Is this how to actually make cross sections of hair and fibers to make them visible? That's also something I would like to try. Yeah. Yeah. Any plans to check on one of the Discord servers? Yeah. I will check because I was very busy the last couple of weeks because end of school, but now the holidays started luckily, so I have a little bit more time. Okay. Yeah, 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 exactly. So I would like to try that method as well to to prepare a slide where you're able to look at the bristles or at hair being vertical from the top. Yeah, so it's very difficult actually to look at that. But there is a technique how to do that, and I would like to experiment with that. Yeah, so maybe yeah, this is something I'm going to try. Do they need sleep? Probably not, I would say. Yeah. I mean, I'm quite sure that they must have some kind of a resting phase as well, but whether they really sleep, yeah. Okay. Um, yeah. So what I'm gonna do now is I'm just going to call it a day again, or an evening rather, because it's already quite late at night of where I am. See, and I think I'm just going to, yeah, wish you all the best and happy microbe hunting, and hope that, as always, that those microscopy sessions also motivate you a little bit to pick up a microscope. Of course, there's always something to discover. Please feel free, of course, to post something in the comments. Yeah, I hope I was able to respond to all of the questions. And yeah, and hope to see you again next week. And if for whatever reason something unexpected arrives and I'm not able to do the live stream, please check the community posts. I will try to activate them a little bit more as well to be in contact with you in case there is some announcements here. Okay, all the best, and yeah, I will check my emails. Okay, happy microbe hunting, and hope to see you next week. Bye bye.